Genome sequence of a 45,000-year-old modern human from Western Siberia. What does modern human mean?
Cro-Magnon? The latest big break in the tree.
Yeah, it means that looks at least looks anatomically like a modern human.
So it's a relative term.
Close enough to us now that they can rule out that this individual was another species. So not Neanderthal, not Cro-Magnon. This is more recently than all of that.
Obviously, that's a perspective question, but so do they base it more on anatomical similarities, or do they base it on cultural things? Why not both?
You'll find out when you read the paper.
Okay.
But one of the things they do look at is anatomy. They get one bone that they're going to, maybe two, I don't remember. One or two bones.
And they're already going from there?
And that's where they start.
That seems quite a jump.
How do they know it's 45,000 years? So the first little bit of the paper is dating the bones. How old are they? How sure are they that it's 45,000 plus or minus how many years? Then they do a little bit on the, is it a modern human bone or is it not? Okay, so that's the, well, read it and yeah, we'll talk about it. That's a good place to start. Do you believe, and so this is, again, this is the whole list of things I could talk about to prepare you for this paper, but we're not going to do all this. So the start of the paper is a lot on how do they know, first two things that the authors will go through in the paper.
Do they tell you specifically what type of isotope they're using?
Yeah, they go into a little bit more detail than I normally see in a genetics paper. Something about collagen, which I could have put up here as a topic as well. Apparently collagen is really important for dating bones. Okay. So this is basically what all of these authors and many other authors are trying to figure out. And we could figure out at what stage in this progression this individual represents. I don't know, maybe something like there, or maybe there. So to make sure everybody's on the same page with background, as we were talking about, we are the only Homo species. There are no other Homo anythings. So what happened to them?
Died out?
Yeah, they've all gone extinct. We're the only extant, currently living Homo species. Of course, we define what's a species. I mean, we could define that chimpanzees are Homo something else. We could, if we wanted to, but we haven't. So all of those other hominins have gone extinct. Here's just a sampling of the different Homo lineages that have gone extinct. So, for example, Homo ergaster, Homo floresiensis, there's the Neanderthals, Homo neanderthalensis, one of the most recent extinctions, with time being 0 present here.
Dang.
We're still living. Neanderthals just died out relatively recently. And then we go back pretty far, almost 8 million years before we reach the common ancestor of us and chimpanzees, Pan. Currently existing non-human primates.
On a semi-related note, did they change Australopithecus, the species, from Afarensis to Africanus?
Or it might be a different,
Just, could be just,
might be two different species.
I guess I'm saying, is it either, oh, it is, never mind.
Australopithecus africanus. According to this figure, which is a cartoon of who knows whose data from a textbook. So don't necessarily believe it. So we're talking about then in this paper Homo sapiens, but just not from now, Homo sapiens from thousands of years ago. They're going to talk a bit about Neanderthals. So I wanted to point out, because I never knew anything about Neanderthals until I started teaching evolution. This was the region of the world where the Neanderthal range was, that is where they lived, where we can find Neanderthal fossils. And the Neanderthal Valley, Neanderthal in Germany, Neander Valley, is in Western Germany. So that's presumably the epicenter, or where the first fossils were discovered. But they ranged about that far. And then this paper is going to refer to two other populations, the Denisova population and Ust-Ishim. And Ust-Ishim is from Siberia. That's where this bone came from, that they've done the genome sequence of this modern, ancient but modern human. Okay, so when they talk about Denisovans, they're talking about fossils that were found in a cave here. Just so you know, this is the perspective. So human ancestor fossils are found all over Asia, China, Siberia, the Middle East, usually cold places where fossils have been pretty well preserved. Textbook.
Which one?
My evolution textbook. I can't see. Norton. I don't remember what the name of the textbook is. What is the name of our evolution textbook? I can't remember. Let's see.
Where'd you get that figure from? It looks really familiar.
You can't read any of this, which is fine. But to understand where these authors are, the perspective they're coming from, there are basically two hypotheses for how modern humans came to be. There's consensus that in either of these two hypotheses, one's called the out of Africa hypothesis, and one is the multi-regional hypothesis. In either of those hypotheses, this multi-regional is colored in this color, and out of Africa, the one that you can barely see, which is not intended. Both of these hypotheses suggest that modern humans evolved first in Africa. The question is, what happened after that? The multi-regional hypothesis suggests that former species, Homo erectus, dispersed all across the world from Africa. So some went to Asia, some went to Europe, some went to Siberia, some went to the Americas. And also some tribes that are in Africa, this center panel, Africa, all of these individuals, all of these groups are African. And then we've got the non-Africans on the side. So the multi-regional hypothesis says that the Papuan from Papua New Guinea, the French, the Han Chinese, Homo neanderthalensis, all went their separate ways from Africa about 2 million years ago. This hypothesis would predict that every human population on the planet is equally related to everybody else. There's a big dispersal. Everybody left Africa and went their separate ways. So everybody should be about equally genetically distant from each other. And notice that what this also predicts is that Homo neanderthalensis left Africa at the same time, about 2 million years ago. They were just unfortunate and went extinct. But this predicts that the rest of the populations survived and are now modern human populations. The out of Africa hypothesis, which is one that a lot of geneticists and evolutionary biologists lean toward at this point, not so much the multi-regional, but the out of Africa hypothesis, suggests that Homo neanderthalensis, like the multi-regional hypothesis, split off, left Africa about 2 million years ago, and then went extinct again relatively recently, about 30,000 years ago or so, just before present. And that modern humans didn't evolve in Africa until about 100,000 years ago. And they stayed in Africa for a while until about like 10,000 years ago or 15,000 years ago, and then one group left Africa. One group of humans left Africa and spread through Europe and then Asia and then the South Pacific. And so that predicts that everybody that's not in Africa is very closely related because they went their separate ways only very, very, very recently in the past. So Europeans, Americans, Russians, Chinese, Japanese, South Pacific Islanders, all very closely related, and that the African tribes, the San, the Yoruba, and other tribes are older because they were in Africa diverging a little bit earlier. So that's the key distinction between these two hypotheses. The one on the right says everybody is equally distantly related to everybody else. We all just went our separate ways from Africa at one point in time. The other one says that happened more recently. That most of the human populations share an ancestor very recently in the past, and that the African groups have an older ancestor. So that's one thing that could be useful for understanding the paper next week. Don't need pretty. Okay. What evidence do we have for this? Phylogenetic tree. Description of the genetic relationships between a lot of extant, currently existing human populations shown here on the tips of the tree. So if you have two, like here in this box, I know you can't read it, it says San and San, two members of the same population. They're most closely related to each other because they're connected by these lines. They're the closest neighbors to each other in the tree. Short distance between the two tips of the tree. So those two individuals are closely related. You have to go way back in time, about 138,000 years, before you find they have a common ancestor with, what does that say? Kikuyu, Hausa, and Mabuti populations. So this is just a description of how closely related human populations are to each other. If I zoom in, you may notice that all of these are African tribes. They all share a common ancestor way back here, about 190,000 years ago. So each of those African tribes have been diverging by themselves over a long period of time. They're all very genetically distantly related to each other. If we look in this part of the tree that's in the top, that share a common ancestor about 83,000 years ago, who do we have here? Basically everybody else. Pacific Islanders, New Guinea, the French, more New Guinea, Chinese, Samoan, Chukchi, Russian, Australian, Dutch, Japanese, Australian Aboriginals, Native Americans, so basically everybody else shares a common ancestor 83,000 years ago.
What is M&M?
Okay, M&Ms are delicious chocolate candy. I'm glad you asked because that's one of the reasons I wanted to show this to you. These different letters, A, N, F, B, N, R, P, J, T, etc., those are just designations for different clades or groups of individuals in this tree. So you can talk about the A clade, which is the Chukchi, the N clade, which is Australians, and so forth. The paper that you read not next week, but for 2 weeks. No, next week? Is this next week's paper? Next week's paper, sorry. My brain's going. Does mention one of these mitochondrial clades. So if you want, when you read that part of the paper, you say, oh yeah, we talked about that. Go here and look up where that letter is on this tree, and you'll know what the authors are talking about. But the important point for background for this paper is that this is one of the things that supports the out of Africa hypothesis. That all of the African populations have very deep ancestry. Their common ancestry goes way, way, way back. But when do all of the other human populations share a most recent common ancestor? About 83,000 years ago, much more recently. So this is the leaving Africa part. There's a group of humans, not modern humans, but ancestral humans in Africa that left about 83,000 years ago and then spread across all of the rest of the world.
Two different clades.
Oh, that's interesting. Yeah, so let me zoom in again.
Northern and southern or something.
Yeah, so we've got New Guinea there, but also one there, and we've got Chinese there and also there. So yeah, that's interesting. So yeah, it could be geographic differences. Those are pretty big. Well, New Guinea's not a pretty big country. China's a pretty big country. So it could be regional differences, or it could be cultural differences. right? Marriage practices in different cultures in China. I'm not claiming to be a historian, sociologist, etc.
Anthropologist?
What? Anthropologist, there we go, thanks.
You're welcome.
I was like, what?
But, I don't get it, because if it's by culture, shouldn't this be by taxonomy? This is, like, it's
Well, all I'm saying is that there could be social, and this is purely hypothetical, because again, I'm not a historian or an anthropologist. If there's a cultural division, like if they're like the caste system, how old is the caste system? It's pretty old. So if marriage is restricted between different members of the same species living in the same region, you'll get very old divergences in lineages. Does that make sense?
Culture functions as a genetic barrier.
Yes, exactly. It's a reproductive isolation barrier. You could have one group. So the same would be true in the US if we didn't have interracial marriage. Right? That breaks down what's otherwise would be what would otherwise known be. Can we go home yet? What would otherwise be known as a genetic barrier to mating. Right.
I actually have
So that could be what's going on. Yeah, go
I have
for
a
it.
question on that same line.
Yeah.
The Japanese, the two of those, I mean, I guess
Yeah.
you wouldn't necessarily know, but is that because there's white Japanese and then dark Japanese? There's some, I've heard stuff about that. Like there was a,
Oh, I don't know.
That one looks like it would fit your caste theory really well.
maybe.
Maybe. Although they are, yeah, so 41,000 years ago,
Right.
but they are close. They're the,
Or it could also be a function of, or at least I believe I've read, with Japan, they had different waves of settlement.
mm-hmm.
People going to the islands,
Uh-huh.
and then later on another group going.
Yeah.
So that could possibly be that. But do you know, have you seen the models for language dispersal?
Yeah, I have.
Do you know how well it overlaps?
Oh, that's interesting. I always wanted to be a phylogenetic linguist or a linguistic phylogeneticist. I thought that would be really
So it was,
interesting.
like, brought it
If
as
I
just
wasn't a
someone-
biologist,
It's like one of them had, like one of the old historians has a very.
mm-hmm. Yeah, how the languages are related to each other. I like it. And then there's this. Oh, go ahead.
Could you go back actually?
Yeah, okay.
I'm just wondering, like,
One of the questions that everybody always wants to answer, and they touch on a little bit in this paper, is did our ancestors have sexual relations with Neanderthals? Which is apparently a very touchy subject. I don't particularly care if my 40th great-grandfather had sex with a Neanderthal, but that's just me. I'm not, my opinion. And then there's this common perception that red hair is somehow a Neanderthal trait. So, just so you know, after Neanderthal DNA started to be sequenced, and I'll go through the quick history here in just a minute. Okay. These authors found that in the melanocortin-1 receptor protein, there's a specific mutation that was in that Neanderthal genome, the Neanderthal gene for this gene, Neanderthal melanocortin-1 receptor. Melanocortin-1 receptor is involved in pigmentation. It's a protein that we all have that controls pigmentation. And so these authors actually put that mutation into a cell line. They made a cell line that has that one specific mutation and wanted to know what does it do, what does that mutation do to the melanocortin-1 receptor protein. And it reduces its activity compared to wild type. So these authors concluded that the Neanderthal version of, if Neanderthals had this version of this gene, which their DNA says they did, then they would have a melanocortin-1 protein that had lower activity than we normally have today. And presumably that would be production of red hair. Light skin, red hair, that mutation. They didn't know that from looking in a cell line, of course, because cell lines don't have hair or pigmentation. But it's that lack of activity that translates into reduced hair pigmentation. But then, so you may at some point hear somebody say, oh, you're a redhead, you're closely related to Neanderthals. I've heard people say that, not to me, but to people that have red hair. And so I want you to know that you can tell them that these authors studied it, and they still concluded that it's not conclusive. There are two hypotheses. One is that Neanderthals had that mutation and passed it into humans. Right? Did Neanderthals have sex with modern humans before they went extinct? Did Neanderthals move any of their genes, their versions of their genes, into the human population? That's basically the question. Are there modern humans walking around that are carrying genes that we got from Neanderthals way back when? And these authors also point out the possibility that it's convergent evolution. That Neanderthals had this mutation and that modern human red-headed people also got it, but that was a totally separate event. It has nothing to do with mating of Neanderthals and humans
It has to be between
thousands of years. So, that's that. Oh my goodness, and then there's this. Was the Neanderthal man going around ravaging all of our women? The real question, which a lot of these genomics and fossil studies aim to answer is, was there geographical opportunity? That is, did Neanderthals and modern humans, or pre-modern humans, live at the same place? Did their ranges overlap? Was there opportunity for mating? Possibly. I mean, that Neanderthal range is pretty big, and as I showed in the previous map, the modern human bone from Ust'-Izem that was sequenced is way up there in Siberia, so the Neanderthal range is between Africa and where that modern human bone was found. So that suggests that humans went through the Neanderthal range when they migrated out of Africa into the rest of Europe, Asia, and the rest of the world. So there probably was geographical opportunity for Neanderthal human mating. Now I want to get really briefly into challenges of sequencing ancient DNA. But first, a little bit on the Neanderthal genome. So there have been many papers. This is the first. Bill Green and a bunch of colleagues in 2010 in Science published the first bit of the Neanderthal genome. And these are the bones that they used. 3 bones from different caves in Europe. And this was their main result, essentially. That French, Han Chinese, and Papua New Guineans carry alleles of genes that were present in that Neanderthal genome. So, barring the likelihood of convergent evolution, that the same changes happened to occur twice, once in Neanderthals and once in all of these human populations, that suggests that Neanderthals and the common ancestor of those groups mated.
What does French entail?
I fear that that might be a question fraught with peril, so I'm not sure how to answer.
Okay, because I know it's like on the chart, it doesn't obviously break down every, like.
Yeah, so I'm not
Spanish,
sure.
Portuguese, all that, so I don't know if French
Yeah.
is general.
Romance versus.
Because I'd imagine it wasn't just.
Teutonic or something.
I would imagine, yeah, it wasn't just France, but neighboring French countries.
Possibly.
Okay.
Yeah, I'm not. I'd have to go back and look at the manuscript, which I have on my computer, so I could.
I have a question.
Please.
So I thought several weeks ago you said that two different species will be considered same species. They can mate and they have offspring.
Yes.
But if modern human and Homo sapiens can mate and have offspring, does that mean that we are the same species?
So this is back. So if this was Neanderthal mating with individuals here, those individuals would also be Neanderthals. Same species, but those Neanderthals were the ones that were the ancestors of modern humans. These ones were going on a different trajectory. So genetically different, but must have been the same species if they could mate and have offspring.
Or subspecies or something.
Exactly. Yeah, excellent question. Yeah, it's all semantics at this point. It's how we define population, species. Yeah. Okay, so then in 2006, these two papers, the one we were just talking about, analysis of 1 million base pairs of Neanderthal DNA, Richard Green et al., and the same, though that was Nature, Science, same year, sequencing and analysis of Neanderthal DNA. So 2 groups were going at this, trying to be the first ones to publish that they could get fossils, take a dental drill and the bones, get some DNA out of the bones and sequence it. That's what they did. They drilled some fossils, they extracted DNA from the dust, they sequenced it. I love what you're thinking right now. So here is, yeah, exactly. Okay, so what are the key points here? This is part of, I don't remember which of those two papers. Although it's possible to recover mitochondrial DNA, this is fraught with special difficulties and pitfalls. So there's degradation and chemical damage, yes. Then there's this really important part. Contamination.
Yeah, exactly. People even digging them up.
Exactly. So humans are out there in the field handling the bones, digging them up. How do we know we're not just sequencing some modern human's DNA? Okay. And then they say this problem is especially severe because Neanderthals and humans are so closely related, you expect to find few or no differences in their DNA versus ours. So how would we figure out if we sequenced the DNA whether or not we were sequencing a modern human as opposed to a Neanderthal? So then they say the first thing we have to do there at the bottom is to find fossils that aren't contaminated with anybody else's DNA. So this particular paper that they do a lot of different things to try to find fossils that have very, 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 very little extra DNA. Then Richard Green later, same first author, a couple of years later, another Science or Nature paper. They get so many of these, but this is really cool science too. So they do the whole genome. So this is the first, those last ones were little bits of DNA from fossils, sequencing a little bit. This is the first time somebody said, here's what the entire genome sequence of a Neanderthal looked like. Neanderthals, by the way, the ones that went extinct, not ancient modern humans, whatever that. This is one of the approaches that they used. How do they know whether the bone is contaminated or whether or not the sequence they're getting out is real Neanderthal sequence? They take all the little bits of sequence and they just ask what genus, what family, what order do those sequences have a best match to of all the DNA sequences that are known at this day and age. And if they don't match to primates, then they're not real Neanderthal DNA. Okay, so 79% of the sequences they got had no hit. No other sequence that's known to man yet looks like those sequences. Could be Neanderthal sequence, or it could be degraded DNA that's falling apart and
Or it could also be other organisms that
could be
got
unknown.
DNA on it that are just no longer around.
Could be other organisms, extinct microbes, yeast, fungi, and that's what most of the rest of these are. Yeast and viruses. Yeast, bacteria, viruses, microorganisms that were on the bone. Contamination. So 8%, 15,000 sequences were primate specific, and they figured those are either human contamination or they're real. So most of the sequence data that they got, they weren't able to use. And that's just one of the problems with doing these sorts of. Okay. How do you do the sequencing? So they're using a specific type of sequencing called Illumina sequencing. It's a specific company that makes a specific machine that people nowadays use to do deep sequencing. That's what this paper we're reading next week, what they do. So this is just a brief overview of how this sequencing works. You start with the DNA you extract on the left there, sample DNA. They probably didn't have to do the second step, fragment it. It's probably very fragmented already. But normally you break genomes into little tiny bits, usually 100 to 1,000 base pairs long. You add these sequencing adapters. So to the ends of all of these pieces of double-stranded DNA, you ligate on pieces of DNA that have a known sequence. You know the sequence, you've defined the sequence, you stick those primers on the ends of the unknown, the part in the middle. Then you go to NGS workflow, to the next generation sequencing workflow. That is, for example, the Illumina HiSeq machine. It's one of their models of DNA sequencer. That's the one that these authors use. These sequencers create just 100 base pairs of sequence. That's all you get. So that's what's in the background here. A bunch of different HiSeq reads from a data set I collected. So here, I'm circling in blue, is one of those 100 base pair reads. That's 100 base pairs of sequence from Caenorhabditis briggsii. The HiSeq machine generates 37.5 billion base pairs of sequence every time you run the machine. Those come in 100 base pair chunks. So I did my math wrong. That's 500 million 100 base pair reads. You get 500 million lines, 100 nucleotides each out of every time you run the machine. That is sequencing the entire human genome 10 times every time you press go on the machine. So you get a lot of sequence. That's why it's called deep sequencing. If you did this for a human genome, you're sequencing every specific base pair in the genome on average 10 times. That's why these are so useful. If you do it 10 times, you get a really good idea of whether or not you're accurate. If you read the same nucleotide 10 times and you always see that it's a T, you're pretty sure that it's actually a T in the genome. Not a G, not an A, not a C. I'll skip this bit. So that was all about how the HiSeq works, but the actual technology is not important. This is important. They're going to be talking, what's going on?
Oh, nothing.
No, what is it?
Oh, the game?
Yeah.
Okay, we're 2 outs away from winning. We're 3-2 still.
Hey. Nail biter, as I said earlier. So one of the things that the paper is going to talk about is mapping reads. I just want you to know, we've talked about this briefly. I've talked about this briefly once. I just wanted to go over it one more time. What does mapping reads mean? So each of these red bits indicates the DNA sequence you got from one fragment of DNA. So the Illumina HiSeq sequenced 2 ends of the same piece of DNA, shown there, so 2 red bits. And then the intervening blue sequence, we don't know, as it says there. 330 to 430 base pairs of unknown sequence in between, but we get the 2 ends of sequence. Then you line them all up like this using a computer. Line up all the sequences, see how they fit together, and that's how you build a genome assembly. So you can find out, for example, that for a specific part of the genome sequence, there are certain sequences here, there, 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 that all overlap. That's how many times we've sequenced that specific part of the genome. 1, 2, 3, 4, 5, 6, 7, 8 or so times. 8-fold coverage. So you can take a little tiny piece of sequence that you get out of the Illumina sequencer from, say, an ancient human, and find out where exactly does it go in the modern human genome. 30 base pairs of ancient human DNA, you just say, where does it fit? Where does that 30 base pairs go in our genome? So you can compare the ancient human genome to the modern human genome. Are there been changes? Is it mostly the same? That's one of the things that they'll do. This is what that actually looks like when you zoom in and actually look at the sequences. Each of these lines represents one read off of the machine, and this is how they all line up. We take all of the reads that resequence the same part of the chromosome and line them up. This is what you get. And since we're all so passionate and good at creating consensus sequences, this is how genomes are sequenced. You've got lots of A's at that specific, which I can't draw exactly. It's all A's. So what do we say the consensus DNA sequence is there, that bottom line? It's an A. Yeah. So that's the power of deep sequencing. As I said, you sequence every single nucleotide that many times, you get really good confidence that you have the correct nucleotide at every spot in the genome. So this is mapping reads and assembling. Are there any nucleotides? There's one here that's got a lot of asterisks, which indicates that half of those reads there's an asterisk. The other half is G. So that could be, for example, a heterozygote. One of your two copies of DNA has a G. The other copy, these scientists can't tell what the nucleotide is, so it's an asterisk.